The plasma membrane is a dynamic structure made of lipids embedded with proteins. In 1972, Singer and Nicholson introduced the fluid mosaic model, which describes the membrane as a two-dimensional fluid, where proteins are embedded within a flexible lipid bilayer. Peripheral membrane proteins bind to the membrane through weak electrostatic and hydrophobic interactions, primarily involving hydrophobic and basic amino acids. Unlike integral proteins, they do not penetrate the lipid bilayer, making their removal easier. They can be dissociated using high salt buffers, for example sodium chloride, potassium chloride, magnesium chloride 2, which disrupt electrostatic interactions with phospholipid head groups, or strong acid and base buffers, which alter charge distribution without breaking the membrane. This allows researchers to selectively isolate peripheral proteins while keeping the lipid bilayer and integral proteins intact. Integral membrane proteins span the lipid bilayer with single or multiple transmembrane regions, making their extraction more complex than peripheral proteins. Since they are embedded within the membrane, they must be dissociated using strong detergents that break hydrophobic interactions, allowing them to become independent proteins. Detergents like Tween 80, SDS, work by binding to the hydrophobic regions of the protein while interacting with the hydrophilic regions, effectively solubilizing the protein without disrupting its structure or function. In a cell fractionation experiment, centrifugation separates cell components based on size and density, where integral membrane proteins typically end up in the pellet since they are embedded in membrane fragments, while peripheral membrane proteins are usually found in the supernatant. And that's a wrap. Membrane proteins may be tiny, but they play a huge role in keeping cells alive and active. If you found this interesting, check out my other videos for more deep dives into cell biology. Until next time, keep exploring the world of biology.